Hello and welcome, dear students. Today we are going to discuss about the topic a brief history of evolutionary genetics. The main objectives of today's lecture are to know the basics of evolutionary genetics, to understand the evolution of the structure of genome, to know the different mechanisms of genome evolution, to know the historical perspective of evolutionary genetics, to understand the synthetic theory of evolution, to know the work of Charles Darwin, and finally to understand the onset of molecular evolution. Dear students, let's start with the introduction first. Evolutionary genetics is a study of how genetics leads to evolutionary change. It involves studying the evolution of the structure of the genome. This is the sum of population genetics and evolution. Genome evolution is the process by which the size and structure of the genome change over time. The genome may develop through mutation, transduction, and horizontal gene transfer. Evolutionary genetics is known as how genetic variation contributes to evolutionary change. This includes the evolution of the genome structure, the genetic basis of adaptation and specification, and genetic change in response to population selection. Evolutionary genetics is the sum of the genetics and the evolution of the population. Dear students, let's know some key points about the history of evolutionary genetics. Exon is the coding part of the gene, that is, exon is a part of the gene expression. The mature RNA messenger consists only of exons, which are later translated into protein. Aneuploidy is a deletion or addition of chromosome number, which in normal humans contains 46 chromosomes, but aneuploidy results in either 45 or 47 chromosomes. Polyploidy consists of more than 2 sets of chromosomes. Transposition is a process in which the segment of the chromosome is transferred to a new position. Dear students, let's discuss the evolution of the structure of genome. Genome evolution is defined as the process by which the genome changes its structure, size over time. Evolution of the genome may happen by a number of methods: mutation, horizontal gene transfer, and transduction. The mutation is any permanent change in the body's nucleotide sequence. Horizontal gene transfer is a method of transferring genetic material from donor to recipient that is different from the vertical transfer, that is from parent to offspring. Dear students, let's understand the different mechanisms of genome evolution. Let's discuss the gene duplication. When a particular DNA region is duplicated, gene duplication can occur through recombination, transposal, aneuploidy, polyploidy, and replication of the slippage, that is DNA replication error. Gene replication via recombination occurs when there is an irregular crossing at the time of meiosis, that is the reductional division or by which the gametes are formed. Let's discuss the transposable elements. A region of DNA that can be inserted anywhere in the genome either by cutting and paste mechanism or by copy and paste mechanism. For example, TY elements in Drosophila fruit fly. The most common transposable element found in humans is the ALU sequence. Now let us discuss the mutation. Spontaneous mutation often cause change in the genome. Mutation induce change in the nucleotide sequence that induce the mutation of the frame shift, thereby altering the genome. It is also one of the processes of adaptation of the genome. Let's now discuss the exon shuffling. During exon shuffling, two exons of two different genes come together to form exon or repeat the same exon. This is the process by which new genes are being created. This adds a new gene to the genome. This plays an important function in the evolution of the genome. Dear students, let's understand the history of evolutionary genetics. Because a genetic population is described as the sum of gene or allelic frequencies for all the genes represented by that population, it follows that for evolution of a species to occur, the gene frequencies of that population must undergo change. The Hardy-Weinberg law describes a population that exists in genetic equilibrium. Several factors can act to change fitness. Viability and fertility are traits that are associated with fitness and are directly related to the ability of an individual to survive long enough to reproduce. By altering the fitness of an individual, the mating distribution will change. The distribution will change because genotypes in the subsequent generation will not appear in direct relationship to the gene frequencies of that population prior to the change. Consequently, the gene frequencies will change and the population will evolve. Dear students, let's talk about the synthetic theory of evolution. The synthetic theory of evolution, as described by Sewell Wright, attempts to explain evolution in terms of change in gene frequencies. This theory states that a species evolves when gene frequencies change and the species moves it to a higher level of adaptation for a specific ecological niche. Several factors such as mutation of alleles and migration of individuals with those new alleles will create variation in the population. Selection will then choose the better adapted individuals and the population will have evolved. The classic example which supports this theory is that of the peppered moth in England. The moth can be either dark or light colored. So prior to industrialization of central England, the light colored allele was most prevalent. The light colored moths would hide on the white bark trees and avoid bird predation. But the pollution generated by the new industries stained the light colored trees dark. Gradually, the light colored moth was attacked and that allele became much less prevalent. In its place, the dark colored allele became most predominant allele because moths that carried that allele could camouflage themselves on the stained trees and avoid being eaten by their bird predators. Clearly, the population had evolved to a higher adaptive condition. Because population change require change in gene frequencies, it is important to understand how these frequencies can change. The three primary methods of change are mutation, migration, and selection. Let's now discuss them one by one. First, the mutation. Mutations are classified as beneficial, harmful, or neutral. Harmful mutations will be lost if they reduce the fitness of the individual. If fitness is improved by a mutation, then frequencies of that allele will increase from generation to generation. The mutation could be a change in one allele to resemble one currently in the population, for example, from a dominant to a recessive allele. Alternatively, the mutation could generate an entirely new allele. Most of these mutations, though, will be determinantal and lost, but if the environmental changes, then the new mutant allele may be favored and eventually become the dominant allele in that population. If the mutation is beneficial, however, to the species as whole, migration from the population in which it initially arose must occur for it to spread to other populations of the species. The most basic type of mutation is the change in a single nucleotide in the gene. Mutations are generally deleterious and are selected against. but the genome of a species can undergo another type of change, gene duplication, which actually favors mutational events. If a single gene that is important undergoes a duplication, mutation in the duplicated copy would not necessarily reduce the fitness of the individual because it still would have a functional copy of the original gene. With this adaptive constraint removed, further changes can occur that generate a new gene that has a similar function in the organism, but may function at a specific time in development or in a unique location in the individual. This type of evolution generates multi-gene families. Many important genes such as hemoglobin and muscle genes in humans and photosynthetic genes in plants are organized as multi-gene families. Let's now discuss the second aspect, that is the migration. One of the assumptions of the Hardy-Weinberg law is that the population is closed, but for many populations this is not the case. Human populations clearly are not closed. Migration will change gene frequencies by bringing in more copies of an allele already in the population or by bringing in a new allele that has arisen by mutation. Because mutations do not occur in every population, migration will be required for that allele to spread throughout that species. Migration in a sociological context implies the movement of individuals into new populations. In genetic context, though, migration requires that this movement be coupled with the introduction of new alleles into the population. This will only occur after the migrant has successfully mated with an individual in the population. The term that is used to describe the introduction of new alleles is gene flow. The two effects of migration are to increase variability within a population and at the same time prevent a population of that species from diverging to the extent that it becomes a new species. The first effect is important because it provides the variability that a population will require to survive if the environment changes drastically. As migration continues over a period of time, the new mutation will be shared between populations. This blending effect helps stabilize the similarities between the populations and prevent more isolated populations from evolving reproductive barriers that may lead to speciation or the formation of species. Let us now discuss the third parameter, that is the selection. A natural result of mutation is that new forms develop, and these new forms may or may not add to the fitness of the individual. If the fitness of the individual leads to a reproductive advantage, then the alleles present in that individual will be more prevalent in the population. In this manner, the alleles of this individual are selected. The process is called selection. In a Darwinian context, this is also called natural selection. These three forces like mutation, selection, and migration lead to change in gene frequencies within a population. But evolution, as defined by Darwin, is driven by natural selection. Dear students, let's discuss about the work of Charles Darwin. Charles Darwin brings forth an evolutionary theory predicated on natural selection. He emphasizes heritable continuous variation within populations, assuming a blending model of inheritance. Darwin's ideas are greeted with fury and adulation, and he becomes the world's most famous biologist. Let's now discuss Gregor John Mendel. Gregor Mendel performs his experiments and formulates his discrete model of inheritance, elucidating the laws of independent assortment and segregation. Unlike Darwin, Mendel's work is ignored and quickly consigned to the irrelevant bin. Dear student, Francis Galton, Carl Pearson, and W.F.R. Weldon forward the evolutionary model via natural selection in a formalized manner using statistical techniques such as correlation and regression. In the process, not only do they delve into evolutionary biology, but they set the foundations for modern statistics. Other workers like Thomas Huxley and more marginal figures like Ernest Haeckel accept the fact of evolution but dissent from or do not extend Darwin's original insight in regards to processes like natural selection upon continuous heritable variation. Though the biometricians around Galton, Pearson, and Weldon continue to carry the torch of Darwinian evolution. Over time, the idea that gradual selection on continuous variation could generate species or drive change was generally rejected by biologists. Before 1900, William Bateson began formulating his own theories which reject the biometrical school's emphasis on gradual evolution and analysis of extant variation in favor of a form of discontinuous saltationism, which ironically Galton himself was favorably predisposed towards. In 1900, Mendelianism was rediscovered simultaneously by multiple workers like Karl Correns, Hugo de Vries, and Teschermark. Bateson became an enthusiastic Mendelian as it was a plausible model for the discontinuous evolutionary process he favored. Dear students, Hugo de Vries and Thomas Morgan were two other prominent scientists who would make their mark as Mendelians. Against them stood Pearson, as Galton was aged and soon passed from the scene, as did Weldon. Within this period, there was a period of defection as workers saw that experiments seemed to favor the validity of some form of Mendelianism, and an eclipse of Darwinism ensued. But in the 1910, a new synthesis emerged as R.A. Fisher showed in the correlation between relatives on the supposition of Mendelian inheritance that Darwinian evolution and continuous traits emerged naturally from discrete Mendelian system via polygenic systems. Dear students, this period was the renaissance of Darwinism, as Mendelianism was absolutely absorbed into evolutionary biology and the groundwork for the neo-Darwinism synthesis established. Central to the process was the work of three theoretical population geneticists like R.A. Fisher, Sewell Wright, and J.B.S. Haldane. Though their specific methods differed, these three workers used formal mathematical systems to elucidate the general principles of evolutionary theory via population genetic frameworks. Fisher emphasized the operation of selection upon large populations. Wright was pluralist, giving more room to stochastic processes, non-adaptive genetic variants, and emphasizing the importance of population substructure, while Haldane tended to approach particular projects in an ad hoc fashion, like genetic load, Haldane's rule, etc., though his general outlook lay somewhere between Fisher and Wright on most issues. The dispute between Fisher and Wright in regards to the salience of parameters like genetic drift and gene-gene interaction gave rise to the Wright-Fisher controversies, which continued for decades and still echo down to the present in their years. While these three workers laid the theoretical groundwork, experimentalists like Theodosius, Dobzhansky, and E.B. Ford set out to test some of the models promulgated by the theoreticians. Ford was an acolyte of Fisher, who founded ecological genetics and showed that the power of selection in natural populations. Dobzhansky focused on the fruit flies to illustrate the importance of population substructure and stochasticity. Naturalists like Ernest Mayr also joined the fray, while George Gaylord Simpson introduced a paleontological perspective. By 1940, these workers had cumulatively generated what would become the neo-Darwinian synthesis, which would be set against macro-mutationist models like that of Richard Goldsmith. Dear students, during this period, workers like Dobzhansky, Mayer, and Simpson were influential in crystallizing the gains of the synthesis. The early emphasis on stochasticity in Wright's model of the shifting balance transformed into a more nuanced system which focused on gene-gene interactions and rugged adaptive landscapes, as well as population substructure. Journals like Evolution were founded, which promoted evolutionary biology as a separate and integrated discipline. In 1942, Julian Huxley published Evolution, the modern synthesis encapsulating the general consensus in the field. Dobzhansky elucidated the difference between the balance and classical schools of evolutionary genetics, placing himself in the balance school against Hermann Muller. The classicals, that is, though one could argue that both were really proxies for the original ideas of Fisher and Wright. While the balance school tended to favor maintenance of extant genetic variation in the form of heterozygosity, often via heterozygote advantage. The classical school argued that homozygosity was the norm, with polymorphism generally being an issue of observed transience, that is, an allele is going to fixation or is being swept to extinction. The extent of polymorphism found practical application in trying to understand the effect of radiation during the nuclear age. Dear students, the discovery of the molecular clock, Colossians, and the two papers on allozyme polymorphism by Richard Lewontin and J. L. Hubby broke the Wright-Fisher balance classical duopoly. Neutral theory promoted by Moto Kimura leapt in to fill the breach, arguing that the high levels of polymorphism and the existence of a molecular clock are evidence for widespread selective neutrality on the molecular level. In other words, transient polymorphism is very common, while over-dominance in the balance school sense is not. A selectionist versus neutralist debate ensues, which goes nowhere, but by 1980, neutrality is held to be a reasonable null hypothesis, and the relevance of selection and neutral processes seem to be important, dependent on context for those not wedded to grand narratives. While the population genetic basis of evolutionary biology was diversified by the molecular dimension, Stephen Jay Gould and Lewontin attacked the Panglossian adoptionism of some thinkers, like that of Ernest Mayr. Simultaneously, a very ambitious form of social evolutionary theory was formulated by W. D. Hamilton, straight out of the Fisherian adaptationist tradition. Dear student, let's know about the onset of molecular evolution. PCR, that is polymerase chain reaction, makes DNA sequencing far more viable. Computers are powerful enough to handle to the new stream of data. Instead of theoretical genes, DNA sequencing allows scientists to conceive of the total discrete digital nature of genetical inheritance. Instead of snapshots of a specific gene, the idea of digesting whole genomes becomes viable. Controversies bloom like 1,000 flowers as theory and data mix in an orgy of activity. The focus on sequencing shifts the limelight to humans as medical rationales drive genome sequencing projects. Microarray technology allows fine-tuned gene expression assays. Dear students, let's now conclude the lecture. The origins of genetics lie in the development of theories of evolution. Evolutionary genetics considers the causes of evolutionary change and the nature of variability in evolution. The methods of evolutionary genetics are critically important for the analysis and interpretation of the massive datasets on DNA sequence variation and evolution that are becoming available, as well for our understanding of evolution in general. Evolutionary genetics is a somewhat redundant expression. After all, Charles Darwin understood that natural selection requires an inheritance mechanism, although it took nearly a century to reconcile his ideas with those for classical genetics. Moreover, much of the reconciliation between Mendelian genetics and Darwinian evolution involved advances in population and quantitative genetics. Dear students, it was all about today's lecture regarding a brief history of evolutionary genetics. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.